Good afternoon, everyone. It is 12:30, and so we will go ahead and get started with our research update webinar. I'm very pleased to welcome you to the first of a webinar series that we plan to hold on a quarterly basis. My name is Brandy Fuhrman. I'm the new Vice President for Research and New Therapies at the Epilepsy Foundation. I joined the foundation in March of this year, and one of the things that I heard loud and clear from across our network is that folks are very interested in understanding what's going on in the research program, what kinds of projects we're funding, what kinds of advances we're accelerating, and what our impact have been. And so this is an effort to be more communicative with the rest of the network in terms of how our research program is performing and the impacts we're having. So I want to thank you all for carving out some time in your day today to spend with us, and I'd like to thank in particular our two presenters, Dr. Angela Birnbaum and Dr. Oren Davinsky. The outline for the session today is that we will do a bit of an intro, and then I'll introduce both of our speakers. Dr. Birnbaum will go first, and after her talk we'll have a few minutes for just some clarifying questions, and then following Dr. Davinsky's talk, I think we'll open it up for further discussion and more in-depth questions. We do have a chat function as part of the webinar, and that's below in the text box that you should be seeing on your screen. If you have a question that you'd like me to ask the presenters, or if you'd like to hold it there for the end when we discuss as a group, please enter your question there in the chat or the question section. And until that time, I'll have everybody on mute just so that we have better sound quality. We are recording the webinar so that we'll be able to post it for folks who are not able to join us today. So the topic that we're investigating today is cannabinoid research for epilepsy, and I think this is an exceptionally timely topic. You're all aware of the importance of cannabinoid research for our community, particularly for our folks who are experiencing uncontrolled seizures and are interested in cannabinoid-based therapies as a new avenue, particularly when they've failed many, many other therapies. And so the foundation has had a real effort to assist in developing legislation across the states and at the federal level that would enable individuals to pursue these therapies with their physicians. It's also timely to talk about this today because the DEA recently released a decision that they will deny a request to reschedule cannabis and cannabinoid-based therapies from the Schedule I category in which it sits now. And the foundation will be issuing a press release very soon expressing our disappointment at that decision. Although the DEA has also said that they will consider expanding the number of growers who could provide cannabis or cannabinoid extracts for research purposes. And so we're pleased about that, but disappointed about the rescheduling decision. I want to remind you all that the foundation also offers a targeted research program on cannabinoids, and actually one of the projects we'll hear about today is a grant funded through this program. And I think it's especially important that as we work to ensure that legislation exists to allow access, that we also support that with research funding to understand how these compounds are working in the brain, how they can be used safely, and what the risks and benefits really are so that physicians can have informed conversations with patients and families. So we are accepting applications for new projects in September, and we'd love to have your help in spreading the word about this program. So with that, I will move to our first presenter, who is Dr. Angela Birnbaum. She is at the University of Minnesota in the College of Pharmacy, and she holds the position of professor of in the Department of Experimental and Clinical Pharmacology, and also the director of the graduate studies in the Department of Experimental and Clinical Pharmacology. She did her postdoctoral training at the University of Minnesota following her PhD at the University of Minnesota. And she has had a real research interest in the pharmacology of anti-seizure medications, as well as their unique effects in special populations, such as pregnant women, children, and the elderly. So she will talk to us today about her project, which was funded through our cannabinoid research program. So with that, I will bring her slides up and move to the beginning and bring Dr. Birnbaum off of mute. Okay, Dr. Birnbaum, please go ahead.
Thank you for that nice introduction. I'm pleased to speak today about our project. The title of our project is Pharmacokinetics and Cognitive Side Effects of Cannabidiol in Adult Patients, and I hope to show you a little bit of what we are going to look at and why it's important. Oh, next. So our overall project goals for this, looking at an objective, is to obtain preliminary data in adults with intractable epilepsy. One aim, we're going to actually look at the cannabidiol concentrations, and we want to look at it during certain conditions. Single dosing with fasting and fed conditions, and I'll show you that in a little bit of why that's important. And also characterizing some of the side effects, and in particular thinking, anxiety, and mood during these individual times. Next, please. So why do our study in the first place? When we looked and wrote this proposal, there weren't really any systematic studies in patients with the cannabidiol in epilepsy. And so the questions were, is it effective, and are there any side effects? And a lot of the studies at the time were being geared toward children. Next. So when we look at a new drug entity or a new indication, when I look at the overall way the drug might work, there's certain questions that I ask. I look at formulations, and are they consistent? I look at the pharmacy aspects. What dose do we give? How and when do I give the dose? And are there certain things that interfere with the dose, such as food effects? And you'll see that in a minute. We look at drug concentration because I want to know how much drug actually gets into the body and is available for the individual. And are there interactions with other medications? Next, please. So when I look at medical cannabis and all those questions I normally ask, and then I look at what do we know, there are a lot of question marks with this medication. We have a lot of states that have a lot of different regulatory approaches to this. And so when we look at consistent formulations and differences as how this might be distributed, such as a whole plant versus an extract, the consistent formulations becomes a very big question. And we don't know the answer to that based on all the formulations that are available over the states. The dose to give, all of these are question marks in my mind and need further study. Next. So the things I think out of that list that we do know a little bit more about, or at least are taking advantage of, is in Minnesota. We do have particular legislation that has made our product a little more consistent than other states. And so we have an advantage here in that we have a pure CBD extract available, and that will allow us to do certain types of studies better than if we had access to a whole plant type of. The regulations are a little tighter. We also have a little bit of information on a food effect, but that needs further explored. Next. So why do I care about concentration and why do I measure that? Well, when I look at medical cannabis and seizure control, I'm trying to find response, which in this case is usually decreased seizures. We'd like to have no seizures. And so I need to know how much of that medication that you're swallowing is actually at the site of action, which is in our case the brain. And so in order for me to get an idea of that, it would be great to take a concentration from the brain, which is very impractical. So we usually use the drug concentrations from the blood, with the patients, our patients are very, very used to doing. Next, please. So overall, in the whole study, we can combine this with other measures to get a more comprehensive picture. And so when I want to look at, is this drug effective in working for us with epilepsy and decreasing our seizure count, we would actually look at things like seizure count to see if it goes up or down as we give the medication. Our side effects, we can do it in different ways. We can have patients report, which is a very usual way to do it. But we can also do more detailed measures like clinicians or in studies. And our study is actually going to look at more detailed measures in things like anxiety, mood, and measure those in a more consistent way. And how to measure, we could do chart review. That's a easy way to do. But drug concentrations tell us more about how much medication is actually in the body, and that can relate and give us more information on what type of doses do we really need to give. Next, please. So of interest, and this goes back to the state regulations, the medical cannabis and the components we're looking at. If your state, such as Colorado, where you're allowed plants and to possess them and smoke them, it becomes different as to what we look at. There's hundreds of compounds in these plants, they're natural products, and with states that allow smoking, you're going to have a lot more compounds in the system that you may not know about or that might be varying from plant to plant. It's very hard to control that consistency in the product. Minnesota has, again, a little bit more about our regulation. It has to be an extracted oil from a plant, and it has to be done in a particular way. We are not allowed to possess plants. We're not allowed to smoke plants. So it helps control that formulation more so that we get consistent doses and are able to do consistent dosing. And that's what makes this study us in a good spot to do a study like this. The two compounds that are in our formulations are the psychoactive drug that you think about when you smoke marijuana, the THC compound, and we also have the cannabidiol, or the anti-seizure type of purified compound. Next, please. Now you might want to know, we know there's a product out there going toward market, and so why look at these products at the state level when we have these other studies out there? And with that, GW Pharmaceuticals is looking toward product indications, but right now patients have access through that through research. And so it isn't something they can go purchase. It isn't available that way. And so they have similar ways to get to it that we do. The indications for that right now are somewhat narrow with the Dravet syndrome and Lennox-Gastaut, which will be more pediatric indications. So we wanted to look at getting more information in adults, and that's a large population that our clinics have. Our two manufacturers, just to give you an idea, again, they have tighter rules. We have 3 to 8 formulations, so we have different combinations of the THC and the CBD compound. But we also have that pure CBD, which makes it a good study product. Next, please. And then again, so that gives us that consistent product. Next. So our question and the way we approach it, besides linking that drug concentration or that amount and indicating what actually gets in the body from that capsule, is by measuring concentration in blood. Next, please. So one of our main questions with dosing in this case was, do we take CBD with or without food? And that sounds like a trivial question to some extent, but because of the chemistry with CBD and THC, it makes them very good candidates for having a food effect, especially with highly fat foods. And then you also have to think about, if you take it with food, do you always take it with the same fat content of food? So there are different questions that come up with these compounds. There has been, this question was looked at by at least one study that showed there was a 10% effect. It doesn't sound like a very big effect. However, if you look at the product, it is a mixed product, THC and the CBD. It also, they were very low doses in the 10 to 20 milligram range. The doses we're looking at in clinic are going to be over 100 milligrams, or 2, 3. They're going to be a lot higher than that. This was also done in healthy volunteers and not patients. Our study is going to be more in adults. They're going to be greater than 200 milligram doses. We're going to compare with high fat and with food and without any food. And then we're going to look at the concentrations as well as the side effects in these, measured in these more detailed ways to measure side effects. Next, please. So the current progress in our study, we've only been approved for a study for a few months, but we have both, I look at it as a laboratory component and a clinical research type of component. In the laboratory, in order for us to measure the concentrations, we have to be able to have an assay up to measure it. This is not easy right now to find an assay for this because it hasn't been that needed. It's mostly in the forensic area where it was being measured, and so it was for a high amount of product, and it's more threshold of types. So in our lab, we actually have a newly purchased piece of equipment that is able to look at the concentrations of this compound at a lot lower levels. And if you want to think about how that relates to anti-seizure medications might be more used to looking at, such as carbamazepine or phenytoin, the concentrations we're looking at here are going to be 100 to 1,000 times more sensitive to that. And that's because of the way the body handles these compounds. And so our assay that we're working on is actually looking at several compounds also. We're looking at that THC as well as the cannabidiol, but we're also looking at 11 other compounds. And one reason for that is we have the ability then to measure concentrations in those individuals that do have the smoke product available or other types of formulations. So we will have that ability also. For the clinical study, we'll be recruiting about 20 patients with completing, we need about 12 patients that have their dose given without any food and then with high fat food. And then we will also study those individuals after chronic therapy, so after they've CBD for a while, to then assess their mood and cognition and their concentrations. Next. Next slide. And this is just to give you an idea of what it looks like when we measure these compounds. This is a chromatogram from our assay, so it's a lot of peaks and a lot of complications, complicated looking. But what it does is it shows that we can, in one blood sample, measure those 13 compounds simultaneously. And that's a big plus for us. So we just have to go through the step once, and we can measure all of these compounds. And I've just circled what it looks like for the CBD and the THC are there. But again, there are a lot of other compounds out there, and a lot more than 13, but that's what we have available as far as standards that allow us to measure. And again, this needs to be 100 to 1,000 times more sensitive than the other anti-seizure medications we're normally working with. As we push doses up, yeah, we can, the concentrations will go up. Next, please. So for the future, after we get the, hopefully, an answer to what does the food effect look like at a higher dose in a patient, we still have other questions we can answer, such as, do we see differences across different doses? That would be a question. Do we need to suggest that we take CBD with food all the time, without food all the time, with particular types of food? And so there's a lot of questions just around food that could be answered. There are also drug-drug interaction studies that we can work toward. These compounds are known to have properties that will make them more likely to have interactions with other medications. And so I see a lot of studies that may be needed for that. I believe that's my last slide.
Thank you, Angela. I want to give people a chance to ask questions about any of the points you made that they didn't quite understand, any clarifying questions. If you have a question like that, would you please write that in the question section? And someone has asked, what is an assay? Oh, I'm sorry, Angela, I think I inadvertently muted you during that. Could you start your explanation again? What is an assay?
An assay is our term for a test. So when you send a blood sample in to measure a drug concentration, we use an assay to measure it.
Thank you. Any other clarifying questions? Okay, then we will move to Dr. Davinsky's presentation. I'll bring that up and make sure that he is unmuted. Okay, so I believe that Dr. Davinsky hardly needs an introduction to this group, but I'll go ahead and give it. He is working as professor in the departments of neurology, neurosurgery, and psychiatry at NYU in New York. He's also chief of service for the NYU Epilepsy Service. He did his education and training, first his MD from Harvard, followed by a residency at Cornell and New York Presbyterian Hospital, and then a fellowship at the National Institutes of Health. And he has been a very prolific researcher in the epilepsy community with nearly 680 publications to his name, quite an impressive number. And I would say that his research interests, he's really helped to put SUDEP research on the map. That's sudden unexpected death in epilepsy, and serves as a principal investigator in an NINDS-funded Center Without Walls on SUDEP, and he also leads the North American SUDEP Registry. And he's also part of our executive committee of the SUDEP Institute here at EF. But obviously his other interest, among many, is to accelerate the development of new therapies and to encourage clinical trial participation and conduct. So with that, I will turn it over to Dr. Davinsky to tell us about the project that he was funded through our New Therapies program, and this provides partial support for some of the studies that he'll tell you about. Dr. Davinsky.
Thank you very much, Brandy, for that introduction. I'm going to review the open-label trial that we and colleagues at multiple other U.S. epilepsy centers did in treating primarily children with treatment-resistant epilepsy. So if we can go to the next slide. So this is an open-label study with Epidiolex, and this is the compound that was referred to. It's 99% cannabidiol. This is the non-psychoactive compound, or one of the non-psychoactive compounds, but the most abundant one in cannabis or marijuana plants. The psychoactive one that is responsible for the high is THC, and that was essentially completely absent or trace concentration. So these individuals in the trial were not exposed to THC, but is a trial only of cannabidiol, which I will abbreviate CBD in some of the slides and my talk going forward. And one of the biggest caveats to highlight is that an open-label study is one in which both the investigator or doctor, as well as the patient, and potentially if they're a child who is aware, then the child or the parent, in all of these cases, if it was a child, the parent was aware that they were actually getting the active medication. And so this kind of study is not the study which would ever be used to get FDA approval. However, these types of studies are absolutely critical to inform us, especially about the safety and potentially to some degree about the dosing, as well as to give some idea if there is some effectiveness in the compound. So this was our open label trial, which was published, actually it's more like 2016, I think it was in December, but officially the date was January 2016 in Lancet Neurology. And there were 11 U.S. centers that participated, a total of 214 patients whose seizures could not be controlled with existing medications, and in some cases, many of these children and young adults also had been treated with dietary therapies, vagus nerve stimulation therapies, epilepsy surgery in some of the cases of focal epilepsy. So it was a very treatment-resistant group. And we looked at the patients who had at least 3 months of continuous therapy as a subgroup in which we felt we have enough data to look at the safety and tolerability. And the vast majority of those, about 80% or 137 patients, had sufficient data for us to include them in the analysis of had there been a change in their seizure frequency from the month-line base before we introduced this medication. The group of individuals treated ranged in age from 1 to 30 years with a mean age of 10.5 years old. The initial dose of the cannabidiol varied a bit based on different centers' preferences, but in general was around 2 to 5 milligrams per kilogram, and with the standard protocol that the 11 centers used, the peak dose at most centers and for most patients was 25 milligrams per kilogram per day, divided in 2 doses. Some centers, including ours, in a selective small number of patients, went as high as 50 milligrams per kilogram per day. And I will say, to echo what Angela said previously, that these are really quite high doses relative to what's being used by many state dispensaries and by some of the Colorado providers that have shipped cannabidiol very widely to individuals in the United States. And they'll often go up to doses of a total of 2 to 3 milligrams per kilogram per day. In some of the studies that are now ongoing, GW and the double-blind studies, we're looking at 10 versus 20 milligrams per day to see if there's any difference in side effects or effectiveness, but that data is not yet available. Next slide. So this just graphically illustrates the flow of people who were enrolled. 214 enrolled. 52 did not have the 12-week of continuous follow-up therapy when we decided to write this study up. 11 had stopped the medication because of different side effects or issues. One had a sesame allergy. The CBD is highly lipid or fat soluble, so you can't put it in water. You need to have it in some type of oil, and sesame oil was used for Epidiolex. So one child had a sesame allergy. One child had an elevation of liver tests. All individuals who had elevation of liver tests were on valproic acid in both the open label and at least in the initial Dravet trial that I was part of in the double-blind trial. And other side effects or lack of seizure benefit led children to withdraw. In one case, there was a SUDEP, which is unfortunately not uncommon in this group of patients with very, very treatment-resistant epilepsy. So 162 had at least 3 months of therapy. 25 of those were excluded because they didn't meet the criteria where we felt we could accurately count the seizures. One was under age 1 year, and that was outside the protocol, so this was a compassionate care situation. 2 had progressive neurometabolic disorders, so it was anticipated that their underlying neurologic condition would worsen, so they were also excluded because they were never meant to be included in the efficacy analysis. And 21 were patients who had only either absence seizures, complex partial seizures, or myoclonic jerks. And the problem with those seizure types is they're very hard to count, especially in children with developmental delays. So as a group, when we were designing this trial, we felt we'd certainly take care of these individuals and offer them the therapy to try to help them, but we would not be able to get reliable data to really determine what the accurate frequency was of their seizures. So we had 137 who were left in which we had data that we felt was reliable to look at whether their seizure frequency was affected by the treatment. Next slide. So this slide, which is a little busy, but it's a summary of the safety and the effectiveness data. The safety, remember, is a larger group. So the seizure types were focal in 36% of those patients, tonic in 41%, atonic in 20%, tonic-clonic in 57%, some type of motor seizure in 85%, and some combination thereof for everybody. In the effectiveness group, it's a little bit less for the focal seizures, a little higher for the tonic seizures, but the other ones are fairly similar. So just to give you an idea of the distribution, these were a lot of the children had Lennox-Gastaut and Dravet, but there were also many young children and adults, young adults, who did have focal or partial epilepsies. Next slide. And this reviews the adverse effects or side effects that were reported in at least 5% of the patients who were treated. And the leading side effect was somnolence or tiredness. Other very common ones were decreased appetite, diarrhea, fatigue, and convulsion. I do personally believe, and I think the double-blind data now supports this, that convulsion is not truly a side effect. These were just children and young adults who had treatment-resistant epilepsy, and sometimes they had a more severe seizure than was typical for them. But for example, in our double-blind study, the incidence of status epilepticus was identical in the placebo group as well as the control group. So I think in cases like this, when you do studies on children or adults with very severe epilepsy, they will sometimes have bad seizure clusters, and that may be part of the natural history of the disorder. Weight increase or decrease occurred in 7% each, and some patients had increased drug concentrations of medication, which I'll get to later, which was considered an adverse effect by some investigators. Among the treatment-emergent serious adverse effects, so these were not just, if someone's tired, that certainly is important for us to note, but it wouldn't be considered a serious one. Of the ones that some investigators, and again, these were determined by investigators, not by the company, and with 11 sites and multiple investigators at some of the sites, there were differences in how people rated things. So as I said, status was rated by some people as a treatment-emergent serious adverse effect. Other people would have considered that part of the natural history and not listed it as such. In 3 cases, or 2%, diarrhea was severe enough, and I think that is a side effect of the Epidiolex. Some of it may be related to the sesame oil vehicle in which it's delivered, was listed as a serious adverse effect. Weight decrease was listed as a serious effect, and 2, and then everything else here is listed in one individual. And again, keep in mind this is an open label study, so we have no idea if any of these effects would be higher in the placebo group, although the double-blind data will hopefully answer a lot of these questions in much more detail. Next slide. And this slide now looks at the efficacy analysis, group of patients and who was in each group. And just to give you the range of syndromes going from highest, which was Dravet, was 20% of the safety and 23% of the efficacy. Lennox-Gastaut was about 20% of each group. And then tuberous sclerosis was about 3%. And then various other disorders are listed as well. Within the other category, which is about 17–18%, include cases with focal epilepsy, which was at least 10 or 15% of the overall cohort. And some of these other syndromes are rare, but different centers had interest. Febrile infection-related syndrome, epilepsy syndrome, or FIRES was a few percent, and those cases actually did quite well, and there's a publication that will come out on those cases. I think there's actually more than the ones in this open label study that were eventually reported in that is in press, and some other rare epilepsy syndromes as well. Next slide. So one of the issues that emerged from our study, which we were concerned about, is that cannabidiol does inhibit the liver enzyme cytochrome 2C19. Now, we all have a set of liver enzymes that digest any foreign chemical, including drugs. This is one of those cytochromes, and we all can have different forms of it. Some of us may be fast at metabolizing drugs, some of us may be slow with these different cytochrome enzymes. But cannabidiol inhibits this enzyme to some degree, and this is the enzyme that breaks apart one of the Onfi or clobazam metabolites, the desmethyl metabolite. And just to keep it simple, taking CBD will raise the levels of this metabolite of clobazam, which is a commonly used epilepsy medication in Dravet and Lennox-Gastaut. And that increase in the levels of this metabolite, in principle, we would expect to increase the frequency of both side effects like tiredness as well as beneficial effects like seizure control. And we looked at this, and somnolence or fatigue was reported in 51% of the patients on clobazam versus 21% of those who were not taking clobazam. So about a significant increase in the risk. The effectiveness, when we look at the greater than or equal to 50% reduction in motor seizures, which were our primary seizure type that we counted, if an individual was on Onfi, they had an average of a 51%, or I should say 51% of them had at least a 50% reduction in those seizures, while on those who were not on clobazam, only 27% had a 50% or greater reduction in motor seizures. So it did seem like clobazam and CBD do have an interaction, and we have seen this with blood levels as well for the metabolite, and that indeed does improve both seizure control and unfortunately increase some of the side effects like tiredness. Next slide. So I will, and just to say the overall, that slide summarized the efficacy data, but it clearly showed a signal. That was a nice response. However, the major issue is that it is not a double-blind study. So I'm just going to give you, in the next couple of slides, some of the publicly released data from the first phase 3 Epidiolex trial in Dravet syndrome. This is a multi-center study done in both the US and Europe. Helen Cross was the lead investigator in Europe, and we enrolled 120 patients across all the sites. In the pediatric age population, 61 were on the drug at 20 milligrams per kilogram and average age of 10, and 59 were on the placebo with a similar age. They were on an average of 3 additional anti-epilepsy medications, and they averaged more than 4 prior anti-epileptic medications. So again, a very treatment-resistant group. And the median baseline frequency of motor seizures or convulsive seizures, which included tonic-clonic, tonic, and atonic seizures, were 13 per month. Next slide. Overall, the experience was that the Epidiolex was well tolerated. Side effects that occurred in at least 10% of the patients on the medication in the active treatment group were tiredness or somnolence, diarrhea, decreased appetite, fatigue, pyrexia or fever, vomiting, lethargy, upper respiratory tract infection, and convulsion. Again, many of these will be similar in the placebo group, but some will be more frequent. 84% of the adverse effects were considered mild or moderate. Among those children who had serious adverse effects, there were 10 in the Epidiolex group and 3 in the placebo group. 8 discontinued due to side effects in the Epidiolex and 1 in the placebo group. So again, I think one important message here is that CBD is a drug. It works in animal models against epilepsy, and we now have clear double-blind data that it also works in at least children with Dravet syndrome, as I'm about to present, but it is a medication and it does have side effects. Next slide. So with regards to the effectiveness data, at the 14-week treatment period, this is what the FDA required of the company, So it includes a 2-week period when the dose was gradually increased and 12 weeks where the child was on the full dose of either the CBD or placebo. And the median reduction in convulsive seizures was 39% in the Epidiolex group, 13% in the placebo group, and it was a statistically significant difference at a 0.01 level. And this is would be considered a good response for many of the medications that are currently on the U.S. market. And keep in mind, Dravet syndrome is one of the more particularly treatment-resistant epilepsies among all the different epilepsy syndromes. Next slide. And I just want to finish on just a plea, and this isn't to you or Epilepsy Foundation specifically, it's more to the government and my colleagues in epilepsy, but there's a huge need for studies of medical marijuana. So the largest group of treatment-resistant epilepsy patients are focal epilepsy, as you heard from Angela before me, and many people in the lay community, I think less professionally, believe that THC is critical for Dravet and other epilepsies to get better seizure control. That may be true, it may be false. I'm agnostic. I think we need to get data to find out. But there are at least 25 states that have now approved medical marijuana. Patients and parents in many of these states are self-titrating different blends of plant material or oils, and we are really kind of wandering in the dark. And as a physician I have people in New Jersey and New York that I care for who are on state dispensary programs, and it's really hard to know, I think, inconsistencies in preparations and what ratio of THC to CBD, if it's helpful at all, remains uncertain. And we need to get systematic data on both the effectiveness and the safety of compounds like THC, since so many children and adults in America are now getting this for epilepsy. Final slide. So to conclude, this is legal in many states, but as Brandy introduced, the federal government still has CBD and THC and any cannabis-based product a Schedule I drug, which makes it in the most dangerous category with no known benefits. And the only way you can do that is to ignore trials for the treatment of spasms and multiple sclerosis, which has led to the approval of THC and CBD in at least 22 European countries as well as Canada. So clearly there's a lot of data. We now have some double-blind data in both Dravet and Lennox-Gastaut that CBD is effective. It's not yet FDA approved. We hope the additional studies will be positive and it does get FDA approved. But I think for both basic scientists as well as patients, lowering the scheduling would make it much easier. We need, I think, a supply of more consistent products across the states. Some states like Minnesota and I think New York do better. States like New Jersey, I think, do poorly using plant material where parents have to make their own oils, which likely does vary considerably from batch to batch. And I think we desperately need double-blind placebo-controlled trials comparing a placebo to both CBD and combinations of CBD and THC. Thank you.
Thank you very much, Dr. Davinsky. While I work to unmute everyone so that you can ask questions, I just want to thank our presenters for sharing the results of their ongoing studies. And it's really gratifying to see that the things that the Foundation has helped to support are really starting to answer some of those important questions like, what is the pharmacology of these compounds? How do they interact with other anticonvulsant drugs? How should they be best taken in terms of, something as simple as what kind of meal should you eat or not eat when you're giving a dose of these medications? What is the right dose for certain groups of our population? And really, how well does this work in different populations of folks with epilepsy, and does it work? Are there folks who we should expect it won't work for? So I think those are really important questions to be answering, and I'm very proud that the Foundation has helped to provide some partial funding to start answering those questions. So I have now unmuted everyone who's on the line. If you'd like to ask a question, now is a great time to do that. If you're not planning to ask a question and you have the capability of muting yourself, please do that so that we have a little bit better sound quality. Are there any questions from folks on the line? Okay, well, while people are thinking up their questions, I can share with you that when I was at the NIH, we did a survey of grants in the NIH portfolio that related to cannabinoid-based research projects, both basic, translational, and clinical. And I personally was very disappointed to find that there's very little that the NIH has funded so far in this area. And my hypothesis is that one of the reasons that we have so few researchers really jumping into the field of cannabinoids in epilepsy is the scheduling. And I wonder if Angela Birnbaum and Dr. Dwinsky, you could talk a bit about your experiences with conducting research using, under this Schedule 1 situation, and what were the, any difficulties that you experienced. Dr. Birnbaum, would you like to go first?
Yeah, sure. The Schedule 1 does create a barrier. We just came from a summit a few months ago where part of the discussion was how to do research on this compound when you have, it's illegal federally, but the states are approving it. And it takes a lot. Some entities don't know how to approach it. Some of the state folks, as well as the institutions, don't know where to put it. If you are measuring it for a lab like ours, if we're measuring it in need product, the NIH has one farm in Mississippi that grows product to where you would get it. There are other ways that we try to handle it, but it can be a huge barrier both from getting a product. Luckily, we have a Minnesota product, so we got around that, but then we still have institutional review boards that want to know how to go forward with this. So there's several types of approvals.
Dr. Dwinsky, any comments on that?
No, I agree. I think the Schedule 1 designation presents an enormous problem, I think, for the sites like ours that used it as a clinical drug. It took 8 months and a lot of paperwork from myself and research associates to get both the IRB, the Drug Enforcement Agency, the New York State, the Bureau of Narcotic Enforcement. These were inspections by multiple officers with guns in their holsters, to do a trial with a compound that is been known to be non-psychoactive, non-addictive, non-habit forming, and which has been consumed by millions of people for 5,000 years or longer. And even worse than that, perhaps, is that one of my colleagues, Dick Chen, who heads the NYU Neuroscience Institute, who's a very eminent neurophysiologist in calcium channels, has an MD PhD student who was made studying the GPR55 receptor, which is a receptor on nerve cells, which is thought to potentially be the way in which CBD works to help control epilepsy. That's not established, but that's what this MD PhD is working on. He was able to finally, after a year, get CBD through all the approvals through the FDA and the DEA, the Bureau of Narcotic Enforcement, and buying a large safe. He was finally able to get it, but by the time he got it and used it, the experiments had failed, and it was probably that the drug was more than a year old and it does not have the longest shelf life. So it just goes to show that this poor MD PhD student was on hold for 18 months, finally did the experiment, and the drug probably failed because it was too old because of the time it took to get it. So I think the Schedule 1 is just a horrific hindrance to scientific study, and this is putting CBD in slices of hippocampus in a petri dish. So it's really the idea of what the DEA is doing to hold up science is just terrible.
Thank you. That's consistent with some of the things I've heard from other researchers as well, that the hurdle of going through to get the approval to have a Schedule I license is itself a barrier, and then once people are in the process of trying to get that Schedule I license, the timing is really onerous. So we did have a question come in from someone who would like to know, so if a person can buy CBD oil in a health store, what is the problem with experimentation with CBD? Either of you want to tackle that?
I'll take a start, and Angela, you're welcome to weigh in too. I think some of the issues are, we don't know, let the buyer beware. There was a FDA letter, which you can Google, that came out now probably more than a year ago, where they looked at 14 compounds that were being sold via the internet publicly that claimed to have CBD, both for humans and interestingly for dogs with epilepsy, which is a common problem with dogs. And I think of the 14 that they tested in a good lab, any of them had CBD concentrations less than 10%. Some of them had absolutely no CBD. So the fact that something is sold in a health food store and says CBD doesn't mean that it necessarily has CBD. And if it says it has CBD, X milligrams, the quality control on that, there's no one regulating it. So it might be perfectly accurate. It could be off by a factor of 10, either up or down. And in some cases, it could also have harmful things. So one of my friends and colleagues, Steve Schachter at Harvard, was studying huperzine, which is a Chinese herbal-derived compound used to treat Alzheimer's, but also showing promising results in epilepsy. And he went to a health food store, and they tested it in the Harvard lab, and the amount of huperzine, these were in major, I don't want to say it's GNC, but some very large health food store chain, varied wildly, 3, 4, 5-fold, and they were supposed to be like 50 or 100 milligrams. So the concentration varied, and some of these derived from China had toxic metals in them as well. So I think there are dangers to using things where you don't know what the source and the reliability.
I have some comments also. It goes, as was mentioned, by consistency of product. And our goal with epilepsy medications is to provide consistent doses so that we have consistency of control. And this is why smoking a plant can be an issue. Depending on where the plant is grown and what the soil looks like, can actually change the composition of plants. When some of these states, New York and Minnesota, they are very particular about when they harvest the plants, the genetics of the plants, all of these things kind of control in a health food store. Part of it was that CBD was being looked at, that was the way it was being used. But now looking
Angela, I think we're having some trouble picking you up consistently. So I think I'll move on to another question that we got from someone, which was, where can we find out more about participation in the research? And I'm assuming that this is in the clinical research studies that you talked about, Dr. Dubinski.
Yeah, no, it's a great question. We get a lot of these questions. Unfortunately, to the best of my knowledge, the GW trials are currently filled up for Dravet and Lennox-Gastaut. The one that will probably be starting relatively soon is a treatment of Epidiolex for children and adults with tuberous sclerosis complex with treatment-resistant epilepsy. Elizabeth Thiel at Mass General is the lead investigator. We're one of the sites. I don't know all the sites, but Elizabeth Thiel would know geographically which sites are involved with this. There are several other companies that have CBD compounds. There's one transdermal, and there's another company that has a synthetic CBD product. I don't know the status of those trials and whether they're recruiting, but clinicaltrials.gov would be a good place to look. and there may be other ones that come out in the future.
Yeah, I just checked clinicaltrials.gov yesterday, actually, for trials recruiting for this indication, and I did not see yet the other companies' trials listed. So I too have heard that they are coming, but they don't appear to be out there yet. And I'll just remind everyone that on the EF website, there is a trial finder tool. You can put in your zip code, and it will return to you studies that are in your near area that are currently recruiting. So I'd encourage you to take a look at that if you're interested in participating, and come back, because those studies are updated as soon as the companies update the CenterWatch website. So I want to give another chance for anyone on the line to ask a question if you have one. Okay, the last question that came in through the chat box was, success in seizure control is a 50% reduction in seizures. Is that correct? That's a great question.
It is a good question. So in the data, there's a lot of ways to report data for effectiveness in seizure control. A greater than 50% reduction in seizures has traditionally been used in many studies for FDA approval as the kind of effective dose that is clinically meaningful. If you give a drug and someone's seizures are better by 15%, that may not be worth the side effects that go with the medication. So a 50% reduction is considered clinically significant in general in clinical trials. For the Epidemics trial, we actually chose, and I think wisely, as the primary outcome measure, the median % reduction in convulsive seizures. And that was in part to be concerned about a placebo response effect. When clobazam or Onfi was tried, if they had done the 50% responder rate, the placebo rate was so high for that that Onfi probably never would have gotten approved. They used median % reduction in seizures, and that was significant, and that was partly influenced the decision by GW and our investigators to go with that. But the 50% reduction is considered clinically meaningful. Obviously, our hope and our goal is to get people completely seizure-free, although that's very hard in people like Dravet who have very treatment-resistant epilepsies.
Thank you for that. Yeah, I agree. And the other reason I thought that this is a great question is because it gets to that, clinically meaningful outcome question. And that really is a question that a greater engagement with patients in the trial design phase can really have an impact on the clinically meaningful outcome that we all agree should be the outcome of that trial. So, as you said, historically, a 50% reduction in seizures has been considered a clinically meaningful outcome, and in some populations that may still be relevant. But in other populations, among our folks with uncontrolled seizures, we may be interested in changing that number. We may be interested in seizure freedom as the clinically meaningful outcome, or some level in between. But this is another place where having greater patient engagement in the research process can, I think, create more meaningful outcomes for patients from our clinical studies. So last chance, any other questions from the group that you'd like to make sure we answer before we close? Yes, go ahead.
This is Jeff Hoyt with the Epilepsy Foundation. Hi, Brandy. Hi, everybody.
Hi, Jeff.
I have a question regarding the CBD oil, the the THC versus the combination therein of having an element of the THC in terms of effectiveness with treating different types of seizures. In other words, I know that some of the cannabidiol that has no THC, in some patients, I think I've heard where there is a degree of THC in there that it's actually more effective when it's done safely in combination. I was wondering if anybody could speak to that.
Well, I mentioned it briefly at the end. So my answer is that I know of many, 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 many parents and patients who swear that they need THC to get seizure control, and that's their observation. And my feeling is, on an individual basis, I fully respect that, and if they have access to their state dispensaries of getting a THC product, I would be supportive that they use it if they feel it's had a dramatically beneficial effect. As a clinician scientist, I am agnostic, and I would say until we know, we don't know. There are many doctors and patients who have sworn and been absolutely convinced that a therapy has worked, and a double-blind study, from surgical therapies to medical therapies to herbal therapies, and when the double-blind study is done, they don't work at all, or they have side effects that are much greater than any potential beneficial effects. So I think until we do the scientific studies, we don't know, but I do think it's reasonable in individual cases that it be made available and watched, and if people do well, they can stay on it. But I think we should work as a society to get scientific data.
Terrific. Thank you, Warren.
Thanks so much for those questions. So we're at 1:30. I'm going to wrap up, but I want to thank you all again for making time in your day to join us, and to our presenters. Thank you very much for sharing your findings with us. And we will look forward to talking with you all again in the October timeframe. Look for emails and alerts from us about our next webinar series. Thanks very much, everyone. Take care.